Hello, my name is Guy Hunt, and today I'm going to talk to you about GPR32 heightened immunosuppressive monocytes in ALS. In today's video, I'll go through a brief introduction to provide some context behind why we're doing this research. Then I'll discuss our methods, the results we've obtained, and the conclusions we've drawn. So, as an introduction, two of the key hallmarks of ALS are neuroinflammation and altered lipid metabolism. And in our research, we're exploring the role of a group of compounds and their receptors which could potentially bridge the gap between altered lipid metabolism and neuroinflammation. And these are called resolvins and resolvin receptors. And resolvins are a group of fatty acid derivatives that help to promote the resolution of inflammation. And they do this by interacting with their receptors, which are called resolvin receptors, which are on a number of immunological cell lines, including monocytes. And one example of these receptors is called GPR32. So because resolvins are fatty acids, they may be affected by the altered lipid metabolism in ALS, and because they work to resolve inflammation, they may also be involved in the neuroinflammatory aspects of ALS. So in our research today, we're going to focus on the role of a specific resolvin receptor called GPR32 in monocytes, and investigate whether there's a difference in expression between people with ALS and healthy controls, as well as within a number of subpopulations of ALS. So in terms of our methods, we've taken blood samples from 40 ALS patients and 20 age and gender matched healthy controls. We then perform flow cytometry on the blood samples to characterize monocytes and identify the expression of GPR32. We then perform a number of pre-processing steps on the flow cytometry data, such as gating on live cells, on parent markers, compensating, etc. And then we perform clustering, mainly to identify groups of monocyte cell populations. And then after we perform the clustering, we test for a difference in the expression of GPR32 within each cluster between ALS and healthy controls, and then also between subpopulations of ALS. So now we'll go through the results we've obtained. First of all, I'm just going to provide a visualization of the different monocyte clusters we've identified. So this is a UMAP plot. Each of the different colors and numbers represents a different cluster of monocytes that we've identified. And on the right, you can see that we've characterized these into certain monocyte cell populations. And I just want to bring out the fact that we've identified a number of HLA-DR negative monocytes, and these have previously been identified as immunosuppressive. And now I'll go through the differential expression analysis results we've got. Just quickly, this plot closely resembles a Manhattan plot where we've got our statistical significance on the Y, so anything above the black line is statistically significant. On the x-axis, we've got our monocyte cell populations. The size of the dot represents the log fold change, and the color represents the comparison we've done. And just in short, we've identified that there's an increased expression of GPR32 in a number of HLA-DR negative monocyte cell populations between ALS and healthy controls, as well as between fast progressors and healthy controls, and fast progressors versus slow progressors. Also, interestingly, if you look at the yellow dots, there's no yellow dot which is statistically significant, and this represents the comparison between slow progressors and healthy controls. And this means that slow progressors more closely resemble healthy controls than fast progressors. So what conclusions can we draw from this? So we've identified that there's an increased abundance of GPR32 in a number of HLA-DR negative monocyte cell populations in people with ALS versus healthy controls. And this is primarily driven by an increased GPR32 abundance in fast progressors. And because HLA-DR negative monocyte cell populations have an immunosuppressive functionality, the increased GPR32 may be an attempt to further reduce inflammation and suppress the immune system in order to combat excessive neuroinflammation in fast progressors. Our results so far provide evidence for the use of GPR32 as a biomarker for disease stratification and a potential new therapeutic target to treat ALS. At this point, I just want to say a big thank you to all of my co-authors, lab mates, collaborators, and funders.